Hello and welcome. I am Tanya Chamberlain from the Division of Anatomy here at the University of Leeds. And in this video we will address some of the fundamental concepts surrounding the study of neuroanatomy. So what is neuroanatomy? Neuroanatomy is the study of the structure and organisation of the nervous system. It is about looking at the brain and understanding its role, and it is about looking at the structure and the function of the spinal cord. But it is also about looking at the slightly less obvious regions of the nervous system, such as those involved with our special senses, like sight and hearing. Let's have a closer look at what neuroanatomy encompasses. We are interested in how the brain and the rest of the nerves work. We will be looking at how these are structured. Like the rest of the body, they are made up of cells, but it is more evident in the nervous system, because a lot of the time we can pull these cells apart to reveal their formation, to understand where these very large cells act. These common tracts of cells are termed neural circuits, and these neural circuits are connections of individual cells to act as a highway to ensure messages are received and processed in the correct place. But we will also need to look at some of the cells of the nervous system to understand how this works. We are not particularly looking at the physiology of these cells, as it is beyond the scope of anatomy, but we will be looking at the fundamental rules that take place because of the specific properties of the cells in question. We will also need to address the blood supply to the nervous system, so we can identify the consequences of vascular compromise. And finally, we'll be looking at the cranial nerves more closely. But why do we need to know about the nervous system? What does the nervous system do? Well, the nervous system is responsible for every function we have as human beings. It is responsible for our moods, the perception of pain, so responsible for keeping us safe. It is also responsible for monitoring our internal bodily functions, such as blood pressure, blood glucose levels, and temperature. It is responsible for monitoring our external environment. What the nervous system does is receive signals, process those signals, and transmit instruction on how to act. That might be to cry, take painkillers, release hormones to return homeostasis, or wrap up on a cold winter's day. So let's have a look at the learning objectives for this video. After watching this and any further self-directed study, you should be able to define the different categories of nervous system, the CNS, PNS, and ANS. Identify the major functional features and divisions of the brain. Identify and describe basic neuroanatomical direction and planes. Define the terms grey matter, white matter, tract, nucleus, ganglion, and cortex. Describe the structural features of the spinal cords. Describe the internal anatomy of the spinal cord. Explain the mechanisms of a basic reflex circuit. The first thing we should do is address the terminology of the nervous system. The nervous system is a huge umbrella term we use to describe all of the brain, spinal cord, and every single of the 100 billion cells responsible for transmitting information. Unfortunately, this is not specific enough for most fields, so we need to subcategorize elements of the nervous system. The first thing we have is the central nervous system. This is composed of the brain and the spinal cord. We also have the peripheral nervous system, which is every other nerve in the body. The PNS can be further divided into somatic and autonomic nervous system. In the simplest of terms, the somatic nervous system is the stuff we have conscious control or influence over, such as the skin and muscles, whilst the autonomic nervous system is the stuff we don't have control over, like the secretion of glands, the actions of the digestive tract, or the vasoconstriction or dilation of the blood vessels. And to further divide the nervous system, the autonomic nervous system can either be sympathetic or parasympathetic. The sympathetic nervous system sympathizes with the environment. So if we are startled, our sympathetic nervous system is responsible for our fight or flight response. It will increase our heart rate, promote increased circulation to our limbs for fighting or fleeing, and increase our breathing rate to flood our muscles with huge amounts of oxygen. Acting in opposition, the parasympathetic nervous system aims to bring our body back to baseline. So it restores our heart rate to baseline, it returns circulation to our digestive tract, and it brings our breathing back under control. It is important to recognise that our sympathetic and parasympathetic systems do not switch on and off even in full flight mode. Our parasympathetic heartbeat regulation acts to prevent the heart beating faster and faster until it dies. My point is, even though we talk about sympathetic and parasympathetic operating at different times, we are really talking about one of the systems having a greater effect than the other, not that one turns off in favour of the other. The nervous system is primarily made up of a group of cells called neurons. There are many more types of cells in the nervous system, but for now we are only concentrating on the cells that transmit electrochemical messages to, from, and within the central nervous system. The first thing we should talk about is that these cells are termed polarized cells, or cells with cellular polarity. That means their structure and physiological properties are different in different regions of the cell. This is important in neurons because cellular polarity means that the signal of the electrochemical impulse can only travel in one direction. There are 3 types of neurons we need to know about. The first one is a sensory neuron. These are also called afferent neurons because they trigger a change in the nervous system. They arrive at the CNS from the periphery and provide the central nervous system with information about what is going on elsewhere in or on the body. The second neuron is a motor neuron, also termed an efferent neuron. This is because they effect a response on the periphery. These neurons exit the central nervous system and transmit a message or instruction from the central nervous system to the periphery. This is often an instruction to the muscles to move in some manner. Lastly, we have these. These are the interneurons, which are located within the central nervous system and act to transmit the afferent signal from the sensory neurons to the efferent neuron in order to precipitate a change. How do cells transmit this information? We won't be going into the depths of physiology during this video, as the role of calcium channels is beyond the neuroanatomy learning objectives. But we will use this as an opportunity to talk briefly about the role of transmission of electrochemical impulses. Here we have 2 very simplified and generic neurons that are displaying cellular polarity. That means they are different at one end to the other. Let's add some labels to this diagram. First up are the dendrites. These might be receptors in the skin that respond to stimulus such as local tissue damage. Then we have the cell body. This contains the nucleus. We have this myelinated axon. These give the cells the white appearance, and this myelination is responsible for speeding up the transmission of the signal. Then finally we have the synaptic terminals at the end of the cell. Let's assume that the dendrites are receptors located in the skin, and these receptors detect damage to local tissue caused by a flame. Once stimulated, these receptors trigger a rapid movement of calcium across the cell membrane, which changes the electrochemical composition of the neuron, and an action potential is generated. The signal travels from the dendritic area towards the synaptic terminals. But why are they called synaptic terminals? Well, that's because the first neuron joins the second neuron via a tiny microscopic space around 20 to 40 nanometers wide called a synapse. This synapse is a tiny space where synaptic terminals come in very close proximity to the second neuron. Let's zoom in on this region. So here we can see that the impulse on this image is travelling from the left side of the screen to the right side of the screen where it reaches the synaptic terminal. It signals to the protein on the cell membrane to release chemicals called neurotransmitters into the synapse. These neurotransmitters, for which there are lots of different types, bind onto the right cell membrane proteins and trigger a change in the electrochemical properties of the cell that generate another action potential. Now, instead of calling these left and right cells as per the diagram, we have more specific names for these cells. The cell before the synapse is called the presynaptic cell, and the cell after the synapse is called the postsynaptic cell. This will be important to remember because when we look at when things go wrong, we can have different symptoms depending on whether it's a presynaptic cell fault or a postsynaptic cell fault. This is the end of our first learning objective, so let's go back and see what we should address next. Now we will move forward and look at a part of the central nervous system and identify the major functional features and divisions of the brain. The brain, like all other regions of the nervous system, is made up of cells. The majority of these cells are neurons, but there are supportive cells as well. We're only interested in the neurons during these videos, as the supportive cells are beyond the scope of this module. The image on the left shows the brain from a lateral position. We can see the characteristic gyri and sulci, or bumps and troughs that are visible. The image on the right shows the medial aspect. This image is taken from a mid-sagittal cut of the brain. This means that the brain is cut along the midline and one hemisphere has been removed. In this case, the left side of the brain to see what we look like in the middle. These two images will follow us throughout this section, so it's important to remember which is a lateral view and which is a medial view. The cerebrum, or forebrain, forms the largest part of the brain by mass. It is formed from an outer part, called the cerebral hemispheres. These are part of the brain that we all think of when we're asked to describe what the brain looks like. We have two cerebral hemispheres that are divided down the midline by a sagittal fissure. If we look at the medial view of the brain, we can see that deep to the cerebral hemispheres we have a region of the cerebrum known as the diencephalon. The diencephalon is part of the inner cerebrum and should be thought of being a bridge between the more primitive parts of the brain and the more highly developed parts of the brain. Let's go and have a closer look at the diencephalon. The diencephalon consists of four parts. First, we have the thalamus. This can be seen here. The thalami are the relay centres of the brain. They act a bit like a switchboard and pass messages onto the appropriate area of the brain where they can be processed and acted on. The next part of the diencephalon is the hypothalamus. The hypothalamus is located ventrally to the thalamus. The hypothalamus is responsible for neuroendocrine and autonomic function. It continually receives transmissions from other areas of the body that monitor blood pressure, body temperature, blood sugar, signs of dehydration, amongst many other bodily functions, and it produces a series of hormones to try and restore the body to homeostasis. Then we have the epithalamus. This sits on the dorsal periphery of the diencephalon. Within the epithalamus is a small gland called the pineal gland. This gland secretes melatonin, which is a sleep-promoting hormone. It therefore plays an incredibly important role in the sleep-wake cycle and maintaining the circadian rhythm. Lastly, we have the subthalamus. The subthalamus is not visible on this image, but as its name suggests, it sits below the thalami. It makes important connections with the area of the brain called the basal ganglia and has an important role in modulating and coordinating movement. Now we have covered the cerebrum, it's time to visit other areas of the brain and look for the part of the brainstem. The brainstem is made up of three parts. We have the midbrain, which is the most rostral part. This area cannot be seen from the lateral aspect, but we can see it on the medial view here. We also have a rounded part of the brainstem called the pons. We can just see this area poking out beneath the temporal lobe on the lateral view, but it is far more readily visible on the medial view. The final part of the brainstem is the medulla oblongata, or medulla for short. This is the most caudal aspect of the brainstem, and it is continuous with the spinal cord. The brainstem houses our vital for life functions. The respiratory and heart rate centres are located in this primitive part of the brain. If something goes wrong with this region of the brain, the results can be catastrophic. The final thing we should look at is the cerebellum. Cerebellum means little brain, and judging by its appearance, it's not difficult to understand how it came to be named this. Sitting most caudally, the cerebellum has an important role in coordinating movements and balance. In patients with cerebellar dysfunction, we often see signs such as difficulty with balance and uncoordinated movement of the legs when trying to walk. That concludes the major regions of the brain. In further videos, we will go into more detail regarding the roles and subdivisions of each area. But for now, let's revisit the learning objectives for this video. We will now look at the neuroanatomical planes and directions, and cover some of the terminology used when observing neuroanatomy. So you'll be aware that I've started using slightly different language to discuss the location of some of the parts of the brain. Previously, we have addressed anatomy with the superior, inferior, anterior, posterior directional terms, but these largely need leaving behind for the rest of topographical anatomy. We use different terminology when discussing neuroanatomy, and I'll explain why in a moment. First, let's have a look at the terms we use. The central nervous system is on two planes. We have an up and down plane, as represented by the arrow travelling through the spinal cord, and we have a back-front plane, as represented by the nose to occipital arrow travelling through the brain. When discussing the position and parts of the brain closest to the nose, we call this rostral. Rostral actually means beak, as in rooster, so this might be a helpful way to help you remember. For focusing on the area of the central nervous system close to the far end of the spinal cord, we call this caudal. That's because caudal means tail, and it is travelling to the tail of the body. So far, so good. We have a beak or rostral by the nose, and tail or caudal travelling down the body like a tail. Here's where it can get a little confusing, but bear with as it will make sense in a moment. The very back of the brain near the occiput is called the caudal region. The top of the brain around the parietal region is called the dorsal brain, as is the posterior region around the spinal cord. The perceivable underside of the brain is called the ventral area, but this is also the anterior spinal cord. So why do we name these so weirdly? Well, humans are rare animals in that we stand upright. We are bipedal, but most mammals in the class of quadrupeds move around on 4 feet. Our evolution into bipedal animals meant that our orientation of our central nervous system is unique compared to most other members of the mammalian class, with a more flexed brain orientation and a vertical spinal cord. So to make it easier when comparing our nervous system with the nervous system of our closely related species, we use the terminology to prevent confusion. This allows neuroscientists, anthropologists, and all those specialists that study the comparison behaviours of animals and humans have a blueprint where terminology can't be confused between the species. Thankfully, we all use the same terms for the planes of the brain. We've already met the lateral view and the medial view, but we will also be looking at the brain from a couple of other aspects too. Here is a coronal view, as then we are looking through the patient's eyes, and we have removed a coronal slice, looking at the cut end. We also have this view. This is a transverse view of the brain. We have cut the top of the person's head off and we are looking down into the brain from above. This view won't be used all that much throughout these videos, but for the sake of giving you a well-rounded view, I thought it best to include it. When we talk about the central nervous system, in particular the brain, we often refer to grey and white matter. To understand what forms the grey and white matter, think back to those neurons we looked at earlier. The neurons are made up of a cell body and a myelinated axon. Can you remember the appearance that myelination gives to neurons? Yes, it makes them white. So the white matter, as seen in the brain, is made up of myelinated axons of the neurons. In the brain, a bundle of these white matter axons that act in a similar manner are termed tracts. Grey matter consists of cell bodies of those neurons. The grey matter is the location where synapses happen. The brain is known for its characteristic wiggles and squiggles on the cortex. If we zoom in, we should see how these are labelled. These are called gyri, or singular gyrus, and sulci, or singular sulcus. The gyri are the mounds, while the sulci are the troughs. If we blow up this coronal section, we can have a look at the grey and white matter. We can see that the darker crust to the cerebral hemispheres. The grey matter crust of the cerebral hemisphere is termed the cortex. The cortex isn't the only place where we have grey matter in the brain. Any other grey matter in the brain is called nuclei. These are sometimes grouped together and named a structure in their own right, such as the thalami, which is just a collection of nuclei. So the cortex and the subcortical grey matter are termed nuclei, and this is where we have the location of synapses between neurons. Whereas white matter are myelinated axons of these neurons, which are termed white matter tracts when you have a bundle of them fulfilling a similar role. A special mention of these white matter tracts should be given. This is a thick bundle of axons that traverse the two hemispheres called the corpus callosum. The corpus callosum allows the two hemispheres to exchange information. It would also be a good time to briefly mention ganglia, or singular ganglion. Neurons that are part of the peripheral nervous system also house their cell bodies somewhere. In the peripheral nervous system, cell bodies of neurons are housed in ganglia. Some ganglia are the location of synapses, some are not. It is not important for you to be able to list every ganglia, with or without synapses in them. That will happen in due course and only where it's relevant. What it is important that you know is that neurons in the peripheral nervous system house their bodies in ganglia. Right. We have covered the brain part of the central nervous system. Now let's have a look at the spinal cord and the mechanism of a simple reflex circuit. The spinal cord forms the second half of our central nervous system. In the human, this vertical cord of neurons travels caudally from the medulla of the brainstem until it is level with the first lumbar vertebrae. The spinal cord gives off branches called spinal nerves at each vertebral level. We can only see the dorsal ones, but we'll take a better look at the ventral ones from another angle in a few moments. The spinal cord sits within a space called the spinal canal. This is a channel made up of spinal vertebrae to protect this fundamental and yet fragile structure. At the most caudal aspect of the spinal cord, it terminates at the level of L1 or L2, and any remaining spinal nerves continue to the bottom of the spine as something called the cauda equina. This is because they are reminiscent of a horsetail. The spinal column is made up of 33 bony vertebrae. As the cord passes through the bony canal, it produces a spinal cord segment that is named after the vertebra in which it arises. There are a couple of exceptions to this rule, but we don't need to look at the ins and outs of it at this stage. There are 8 cervical segments named C1 to C8, 12 thoracic segments called T1 to T12, 5 lumbar segments called L1 to L5, 5 sacral segments called S1 to S5, and a single coccygeal called CO1. Each of these segments gives rise to a pair of spinal nerves. One nerve goes off to the left and the other goes off to the right side of the body. In order to get a true appreciation for the complexity of the spinal cord, let's have a look at this transverse section of the spinal cord taken from the thoracic region. The top of this image represents the dorsal aspect of the spinal cord. This is the area closest to the skin of the back. And the ventral aspect of the spinal cord is closest to the inside of the body. Like the brain, the spinal cord has a mixture of grey and white matter. The grey matter of the spinal cord is represented by the dark grey portion of this image, whilst the white matter is paler in comparison and forms the perimeter of this image. In the spinal cord, a chunk of white matter is called a white column. This is the dorsal white column. Here is the lateral white column. And lastly, we have the ventral white column. These are paired, meaning that there is one dorsal, one lateral, and one ventral to either side of the spinal cord. But I'm going to keep this image easier to follow. I've chosen to only highlight those on one side. We also have this interesting white matter that crosses over the midline. This is called the ventral white commissure. This allows neurons from the right side to cross over to the left side of the spinal cord and vice versa. We will look at this in a later video when we consider pathways associated with pain and temperature. For now, we will look at the grey matter of the spinal cord. The grey matter are called horns. We have a dorsal grey horn and a ventral grey horn. These are where sensory fibres and motor fibres enter and exit the central nervous system respectively. We also have an additional grey horn called the lateral grey horn. This is only present between the levels of T1 and L2. These lateral grey horns are where nerves associated with the autonomic system leave the spinal cord. If you are looking at a cross-section of a spinal cord without a lateral grey horn, you must be looking at the spinal cord outside of these two windows. The peripheral nervous system must communicate with the central nervous system for there to be a response, whether that be for the perception of touch or pain, or whether that be for an action to result in response to stimulus. If the peripheral nervous system fails to communicate with the central nervous system, then nothing can happen. We will not feel, we will not respond. The peripheral nervous system is made up of afferent and efferent neurons. Afferent neurons arrive at the central nervous system and provide a message to the central nervous system to tell the central nervous system that something has changed in or on the body that requires either recognition or a response. The peripheral nervous system has efferent fibres. These are neurons that exit the central nervous system and instruct the body to act on information that has been provided to the central nervous system. To summarise that, sensory information arrives via afferent neurons, and the CNS instructs the body to act via efferent neurons. A closer look at the spinal cord will help us understand this process a little better. Here are two spinal cord segments. We will zoom in on one of these spinal cord segments and have a look at the spinal nerves. Remember, each spinal segment gives rise to a pair of spinal nerves. From the ventral aspect, we can see a collection of motor or efferent fibres exiting the spinal cord. These are termed ventral rootlets. On the dorsal aspect, we have the dorsal rootlets, where sensory information or afferent fibres enter the spinal cord. Ventrally, these rootlets come together to form a ventral root. Dorsally, these dorsal rootlets come together to form a dorsal root. Just next to the dorsal root, we can see a swelling that isn't present on the ventral root. This is called a dorsal root ganglion, and this is where cell bodies of the sensory or afferent neurons are located. There are no synapses in these ganglia. Finally, the afferent fibres and the efferent fibres, dorsally and ventrally respectively, come together and create a spinal nerve. So the spinal nerve is able to carry information to the central nervous system and also carry instruction from the nervous system. And this is where we can see it happening. Sensory information is carried to the central nervous system by afferent neurons. If you look at the dorsal root ganglion, you can see where the cell bodies of these afferent neurons sit. The afferent neuron synapses with an interneuron in the spinal cord. Specifically, it synapses in the dorsal grey horn of the spinal cord. That interneuron then synapses in the ventral grey horn with an efferent motor neuron, which takes the instruction from the central nervous system to the muscles to act. This is called a basic reflex arc. This kind of communication between the peripheral nervous system and the central nervous system is reserved for rapid response to a stimulus that could potentially cause harm to the body. This reflex arc isn't used when we are deciding how to respond to a stimulus, but is instead used without there any delay by the brain in times of potential tissue damage. Let's have a look at a situation in which this might be used. Here we have a flame. Flames can be dangerous to tissue. It is only here to represent something hot. You could substitute this for a hot pan or a set of hair straighteners or anything else that has an immediate risk of damage to the skin if grabbed. Upon immediate damage to the skin, receptors in the skin form dendrites of afferent neurons fire an action potential that transmits this information of this sudden change of temperature from the skin to the central nervous system. Within the dorsal grey horn of the spinal cord, afferent neurons synapse with an interneuron. This links the afferent transmission directly to an efferent neuron. The interneuron synapses with the efferent motor neuron in the ventral grey horn and generates an action potential to the efferent neuron to send the message to the muscle of the arm. As a result of this, the arm contracts to remove the hand away from the danger of the stimulus, in this case the flame. This is a basic reflex arc. We have evolved this response to prevent damage to our body. It removes the time taken for the message to travel up to the brain to be processed by the brain, for the brain to decide to instruct the muscles of the arm to move, for the message to travel back down to the arm, and then for the muscles to contract. This, so to speak, cuts out the middleman, and therefore the time that the hand has in contact with the dangerous stimulus, preventing excess damage occurred to the skin of the fingers. This is not how programmed movement occurs, but how the body has evolved to adapt to dangerous and potentially damaging situations. And that brings us to the end of this video. We have now covered the learning objectives. With a bit of revision, you should now be able to define the different categories of nervous system, the CNS, PNS and ANS. Identify the major functional features and divisions of the brain. Identify and describe basic neuroanatomical directions and planes. Define the terms grey matter, white matter, tract, nucleus, ganglion and cortex. Describe the structural features of the spinal nerves. Describe the internal anatomy of the spinal cord. And explain the mechanism of a basic reflex circuit.